Hello, YouTube. It's me, David. I haven't posted a video since a long time ago. I think it was like AP Bio. But here we are again. We're going to be covering a totally different thing than AP classes now, because we're graduating from that. We're going to be covering the MCAT. We all hate it as pre-meds, but it's something that we have to take. And so now I'll be covering a single passage from the AMC that has graphs and data interpretation. This is made in response to a Reddit post asking about how I go about doing analysis involving graphs. And it is something that's tricky. If you haven't had a lot of experience doing it, it can be hard. And that's understandable, but the more practice you do with these problems, the better you'll be at them. But here, I will give you an introduction on how I approach these kinds of problems, and hopefully you might learn a thing or two. So, let's begin. Alright, so, I'm going to be covering passage 2 of the section bank. If you haven't done this part, I recommend either just not watching this video, or if you just want to get a learning experience on how to approach a single passage, there's still over 90 other problems to practice from, so it's up to you. So of course, first thing you need to do is read the passage. It's important. The passage has most of the critical information that you'll need to answer, often the majority of problems. There are certain situations where the questions are majority discrete in that they don't need the passage's information, but there are also questions where pretty much everything is based on the passage and nothing is based on just outside knowledge. So we'll see about passage 2 what it's like. So, the passage goes, in the human intestinal tract is inhabited by a diverse assortment of microorganisms known as the gut microbiota. These microbial populations are increasingly implicated in the regulation of host metabolism and development of obesity and diabetes. The catabolism of dietary fiber by gut microbiota result in the production of short-chain fatty acids, acronym, such as butyrate and acetate, which are important energy sources for the host. Acronym are also act as signaling molecules with the ability to bind and activate G-protein coupled receptors, GPCRs. The GPCR43 is expressed in white adipose tissue, WAT, and is not expressed in muscle or liver. Researchers generated GPCR43 deficient, negative GPCR43 mice to analyze the contribution of GPCR43 to energy regulation. Wild-type WT and GPCR43 negative mice were fed a standard diet of STD or high-fat diet, HFD, and their body weights in grams, G, were measured weekly for 16 weeks. Figure 1. So, this is the figure. Let's quickly go through and try to understand what's going on in this figure, because there definitely, most likely, will be a question about this figure in your question. So, a quick glance, we want to see what information we can glean from this. So, we see that there are four different things that they're picking from. There's the wild type, the GPCR43 negative. These two are the standard diets, so basically a regular diet. And then, once again, the wild type and the GPCR43 negative, but now the high-fat diets. So let's see what each one corresponds to. If you have the wild type with the standard diet, you see that the body weight increase is the lowest. If you do the GPCR43 negative of the standard, you see that it's a little bit increased, but it's still relatively low. But now when we go to high-fat diets, you see that both of these are quite a bit elevated in fat. Specifically, though, the GPCR43 negative is the highest. It is the most fat. So this probably is indicating the GPCR43 is somehow implicated in the development of adipose tissue. We don't know exactly how it's implicated, but it seems to indicate that GPCR43 is important to it. Because you see both for the regular diet and the high-fat diet, the lack of GPCR43 increases fats. So that's a general premise of this graph. Next, let's continue with the passage. Next, researchers examined the relationship between GPCR43 function and insulin sensitivity. Adipocytes from wild type and GPCR43 negative WAT were incubated in the presence and absence of both insulin and acetate, and the uptake of radioactive glucose was measured. So this is basically a paragraph describing probably what they did in this figure, graph 2. So let's look at graph 2. First, when you're looking for these types of bar graphs, you should always be looking for something that's significant. Often, significance is indicated with this asterisk, but you can also determine significance if the bars do not overlap. If this bar does not overlap with that bar, it indicates it's significant. In this case, you can see that the bars probably do overlap, so not significant, not significant, not significant. But here, you see that there's something significant. So that probably indicates something's important going on here. But, regardless of that, let's just look at the graph in total and see what we can glean from this. Also, reading the label can also be helpful. So you see that the figure 2 is labeled uptake of radioactive glucose in wild type and GPCR43 negative adipocytes in the presence and absence of both insulin and acetate. Note that the asterisk indicates a p significance value of less than 0.05. Remember that 0.05 in statistics is usually the thing that people care about for whether or not something is significant. So, let's see what this first one is. We can see in this first one, the radioactive uptake of glucose, the comparison is in a wild type, where there is no insulin for both, but the second one has no acetate or acetate. You see that introducing acetate doesn't do anything for the wild type. Similarly, if you do the GPCR43 negative, introduction of acetate also doesn't do anything. But you notice that in the wild type, when insulin is present, the introduction of acetate causes significant reduction of radioactive glucose uptake. So what does that mean? It's probably telling you that the acetate causes a reduction of glucose uptake. So it seems to be counteracting what insulin does, because you know that insulin tells cells to uptake glucose. As you can tell, normally there's an increase in glucose uptake. You can see this by comparing like the wild type negative versus when the insulin is present negative. You can see that's reduced. But interestingly, once again, the acetate isn't doing that. So we probably are giving information that the acetate is important to how insulin functions. And then, interestingly enough, when the GPCR43 negative is here, the introduction of acetate doesn't do anything. It doesn't cause a significant change. The insulin is still doing its effect of increasing uptake. So this, we can probably glean information that the acetate is only able to modulate insulin's effect when the GPCR43 is present. When it's not, it can't modulate. So that is how you go about going through the passage and understanding what's going on. So now, let's move on to the actual questions. First question of this passage is, which table shows the expected body weight of wild type and GPCR43 mice fed a high-fat diet while housed for 16 weeks in a conventional con condition, or germ-free GF condition where the gut does not become colonized? Alright, so what is this question telling us is going on? We need to understand. So first, they want us to know what's the difference between the wild type and GPCR43 negative. You can see that each one of these tables is wild type GPCR43 negative. Next, you see that they're all going to be fed a high-fat diet for 16 weeks. So everything here you should understand is being fed a high-fat diet. But now there's an additional condition for the wild type versus GPCR43 negative. They could either be getting conventional condition or a germ-free condition where the gut does not become colonized. So the conventional condition would just be what's going on, probably similar to what's going on in figure 1. To the not standard diet, but just like what happens regularly for a high-fat diet, which you saw was an increase in fat. But what's different about a germ-free condition? Let's think. Going back to the passage, remember that the passage was talking about gut microbiota. So maybe there's something in the passage that's helpful here. And there is. Notice that this passage specifically tells you that microbial populations are increasingly implicated in the regulation of hope metabolism and development of obesity and diabetes. So, this is indicating that the gut microbiota does something. Going further, you can see that catabolism of dietary fat by gut microbiota results in the production of short-chain fatty acids, such as butyrate and acetate, which are important energy sources for the host. These molecules also act as signaling molecules, which bind and activate GPCRs. Specifically, they bind the GPCR 43. So, if these bacteria are not present in the gut, they can't be making the butyrate and the acetate, which means they can't be affecting the GPCR, right? Because the GPCR only will activate when what? When there isn't, or when there is acetates or butyrates. They need a signaling molecule. But the fact that there is no gut microbiota means that there is no signaling molecule. So, in effect, what does not having a gut microbiota have? It probably has a similar effect to having a GPCR 43 negative on a high-fat diet, because there's no signaling molecules to activate this, so the fact that it can't be activated is effectively the same thing as not having it at all. So we're looking for an answer that causes you to see a reduction of fat when sorry, I just got trained. We're going to be looking for a reduction of fat when we're in a conventional diet, but the regular germline, the germ-free, will still have an increased level of fat. So, let's look at A. Wild-type conventional GF. Notice that these are about the same. Their body weights are about the same. Is that what we're expecting? I don't think so. Why? Well, the conventional will have the microbiota. So the microbiota should be centering out the butyrates and acetate molecules. They're telling the cell to not increase in weight. So this weight should be significantly less than the GF. But maybe this question is still right because it is a little bit less. So maybe. Don't completely rule this out yet. But these seem pretty close. So in reality, this is probably within the statistical margin of error. You'll need to look at other questions to confirm that, though. Let's also look at the GPCR 43 negative. You notice that in this case, it's 52 and 43. We can rule this question out now. Why? Because we know when the GPCR 43 negative is present, we should have an increased level of body fat compared to the wild type. Remember. Or, not that there would be an increase, they should be the same as the wild type. Because both of them are being fed a high-fat diet. So this one should be about the same as the wild type. These should both be around 60. They're not. They seem to be lower. Which doesn't make sense, because remember the high-fat diet should cause the same amount of fat. So this doesn't make sense. Throw this out. What about B? We have a wild type, we have a conventional, and we have the gut microbiota free. We see that the body weight for the conventional is 40, and the other one is 58. I think we're on to something here. The conventional here has less body fat than what is going on with the gut microbiota free. And this makes sense, because why? With the butyrates and acetates present in the wild-type conventional, we should be having a suppression of fat uptake, which corresponds to the wild-type high-fat diet, which has lower fat than the GPCR43 negative high-fat. So you can see that this is less. But we're not done yet. We still need to look at the GPCR43 negative. You can see that the conventional and the gut-free microbiota both have about 60 again fat. And this makes sense. Because since the GPCR43 is knocked out in this case, it is equivalent to the GPCR43 negative of figure 1, which is just the highest amount of fat. So we see the highest amount of fat. It's around 60, just like the other one, as a gut-free microbiota. So this should be 60. And once again, if you have a gut-free, now you don't have the signaling molecules or the GPCR. So in effect, you're just doing the same thing twice. You just don't have the GPCR43 being activated. And it's not even present. So once again, elevated level fat. B, I think, is the right answer. So this is most likely the right answer, but you would still want to go for the other answers and make sure that they're not correct. Let's look. Wild type, GPCR43 negative. Okay, these both have a reduced amount of fat. This doesn't make sense. I've been saying that the conventional wild type should have less fat compared to the gut-free microbiota. So this should be increased, or this should be decreased. So rule this question answer out. What about this one? The wild type, the conventional GF, we see in this case, the gut-free microbiota has a reduction of body fat. That doesn't make sense. The gut-free should be no signal molecules of butyrate or acetate that causes a reduction of body fat. So this one should be increased compared to that one. Since it's not, this is probably not the right answer. So let's confirm this answer and go on. And you see, it was the right answer. We can see that the conventional does have less, and the gut-free is increased. Let's make sure that their answer, what they choose is the answer, my explanation is correct, basically. So this is a biology question that falls under the content category, structure, and integrated functions of the main organ system, blah, blah, blah. The answer to the shown, blah, 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 gain more weight than a wild type. This is the receptor functions. Yep, so you see, they're talking about the same thing. This suggests that this receptor functions to suppress fat accumulation. That was what I was talking about. Since this receptor is activated by SCFAs produced by the gut microbiome, when mice are grown under germ-free conditions, they do not activate the GPCR. Exactly. They're saying the exact same thing. If you don't have the gut microbiota, as stated in passage 2, you don't have the short-chain fatty acid molecules, and so you don't have the ability to suppress the fat accumulation. And so they should be gaining more fat than when they're under normal conditions. And once again, the GPCR43 mice would gain more weight than the wild type regardless of the presence or absence of SCFAs. Why? Because if they're present or they're not, it doesn't do anything because you just don't have the GPCR43 at all. So you can't ever cause this signal to reduce fat uptake. And so yeah, that's how you go about answering the first one. And so this required you to both use the passage and understand why gut microbiota are important, and it required you to understand what was going on in figure 1. Notice this question required no external information. The passage alone had everything you needed to be able to answer this question. Would it have been helpful if you happened to have done research in gut microbiota and done this type of thing? Yeah, it would have been helpful, but you didn't need it. You didn't need to have studied this at all to be able to answer this question. All you needed was to understand what was going on in the passage. All right, let's move on to the second question. Which amino acid is least likely found in one of the transmembrane domains of GPCR43? All right, straight up, this type of question is a pseudo-discrete. It's like a discrete problem near the end of the section, but it's in the middle of a passage. This question has very little to do with the actual passage. You need to just do this based on external information. So let's see. They're about aspartate, glycine, tryptophan, and phenylalanine. Okay, so let's look at the amino acids and think about it. All the amino acids have, you know, the NH and, God, it's been a while. Sorry, I haven't. I think there's an R group here, and then, you know, the carboxylic acid. This is always the same. So this part, we don't care. What we care about is the R groups. What is a spartate's R group? A spartate's R group corresponds to some sort of something like this. It has the negative charge. Although, what is it? There should be 2 carbons. So if we say, okay, this is 1 too long. So if this is the R is here, 1 carbon, 2 carbons. That's our spartate. Remember, the glutamic acid is similar, but it's just 1 chain longer. So the glutamate one would have been this one. Okay, so immediately I know the answer is probably A. Why? Because this is a charged molecule. It's charged, and it's not going to want to be in the middle of the system. Why? Because the transmembrane domain is the area where it's a bunch of fatty acid, the phospholipid bilayer. It's this area where you have all these long chains of carbons, which are nonpolar. So the intermembrane domain is going to be pretty nonpolar. You're not going to want to put something charged in the middle of this. It's not going to want to stick here. It's going to want to stick on the outside where all the H2Os are floating around. So it's probably A. But let's look at the other ones. Glycine. Glycine has an R group of just H. So this could probably be in the middle of the membrane. It's less likely than some really nonpolar things, but it still could be. I mean, an H is nonpolar, so yeah, could be sitting there, maybe. What about tryptophan? So tryptophan has this kind of indole group. So what's an indole? It's this 2-member ring structure that has an NH, something like this. 5-member ring plus a 6-member ring. I think shoot. I think it's something like this. Something like this. No, we're sorry. This would be too many. Remember that you can't have 5 double bonds. So maybe something like no, there's not a double bond here. So maybe something like this. Where, okay, let's redo that. Nope, that's a 6-member ring. We want this 5-member. So maybe something like this, and there's still the H here. Maybe. I'm not 100% sure what the exact indole group is, but it's going to look like something like this. Regardless, you should immediately be able to tell this is going to be something pretty nonpolar. It's going to want to stick in the middle. I mean, this big benzene ring, and then this thing, I mean, the nitrogen's maybe a little polar, but this overall structure has a bunch of carbons, so it's pretty fatty. It's going to want to be in the middle. And then finally, what's the other one? Phenylalanine. Okay, well, phenyl tells you that there's going to be this ring in it, because these are phenyl groups, right? And then alanine. Remember that alanines are basically just 1 carbon up. So if we just have a regular alanine, it's going to be something like this. With 1 carbon coming off of it. But it's a phenylalanine, so it's these 2 things combined. So what it actually is probably something like this. So alanine, and then the phenyl group attached to the alanine. Sorry for how bad these are drawn, but you get the point. Phenyl group plus alanine. Once again, this is a big non-polar thing, we'll want to stick in the middle. So the only one that is the best answer would probably be the aspartate, because it's charged and is polar. So let's check. Yep. So this question is a pseudo-discrete problem. We didn't need to do the passage to be able to answer this question. All you need to know is what the amino acids look like to be able to answer this question. Alright, question 3. Compared to wild-type mice, which experimental group of the mice is most likely to remain lean when fed a high-fat diet? Well, this is probably just understanding the figure 1 again. So, which one's most likely to be fed a high-fat diet? So, a mouse treated with antibiotics. Okay, what do antibiotics do? They kill bacteria, right? Well, what's the gut microbiota made of? They're made of bacteria, right? So, if you're introducing something that kills bacteria, you're probably killing the gut microbiota, which are making those short-chain fatty acids, which are used as a signal. And from the first question we did, we know that those signal molecules are important to GPCR43's activation, which is important to causing fat suppression. So, A's probably ruled out. Not a good idea. So what about mice treated with a GPCR43 antagonist? Alright, so this is some definitional terms that you need to understand. What is an antagonist? It's something that inhibits whatever it's binding to. In this case, it's going to be inhibiting the GPCR43. And what do we say about the GPCR43? We say that the GPCR43 is important to preventing fat accumulation, as you can see from figure 1. GPCR43, important. Wild type versus GPCR43 not present. When it's not present, it has an increased level of fat. Once again, also for a high-fat diet, an even more pronounced difference between the two. So this is also probably not a good idea, because you don't want an antagonist that's suppressing the GPCR43's activity. Not a good thing. Alright, what about C? Mice in which GPCR43 is overexpressed in WAT. Now that's interesting. This is not something directly in the figure, but now we're doing something where the GPCR43 is overexpressed. So let's think about it. In all these cases, we've been talking about the suppression of GPCR43 with a knockout. So what if we do the opposite? If we increase the GPCR43, we're probably going to have, what? Enhanced fat suppression, right? That's most likely what's going to happen, because now we have more GPCRs, so the signal molecules activate more GPCR43s, which means we get a stronger downstream effect, which means more fat suppression. So this is likely the answer. When we introduce an overexpression of GPCR43, we're probably going to be getting an increase of fat suppression. But let's just make sure. What about D? Mice treated with a drug that inhibits the generation of intracellular second messengers. Okay, so what are second messengers? Remember that when you have the GPCR signaling... Whoops, that's not what I wanted. Let's think about it. We have the GPCR, it's on the cell surface, and then we have the GP whatever. You know, the things that go to it. Remember that we have something binding to it. Let's say, I don't know, we'll say this is the butyrate. When it binds to the GPCR, causes the G proteins to change, causes signals, blah, blah, blah. What's important is it causes these things called second messengers. These second messengers carry out further signaling, which enhances the signal, and can cause further second messengers or the end target and to activate. Or it keeps causing more second messengers to keep cascading down and down. The general point is that the second messenger is important to cause the signal transduction. So, what would happen if we introduce a drug that's inhibiting the generation of intracellular second messengers? What the effect of that is we're effectively killing this section of it. Well, what is the effect of killing that section? The signal's not going to be sent out, right? So that's probably not good. In effect, we're having the same effect as if we just killed GPCR itself. So, killing this would also cause a message to not be sent, and so fat suppression couldn't be carried out. So, C is most likely the correct answer. And it is. Let's just make sure that their answering is similar reasoning. This is a biology question that falls under the content category, transmission of genetic information from gene to protein. Yep. The answer to this question is C, because increased GPCR activity would be expected to compensate for increased fat intake and prevent fat accumulation. Exactly. They're talking about the same thing. If you increase the GPCR 43, in effect, you're enhancing the signal. So enhancing the signal means more fat suppression. And so you see this question is both an integration of understanding outside information and the passage. You need to understand what the GPCR 43 does, and you know that from the passage. But you also need to know critical terminology. What's an antagonist? You need to know what that is. And you also need to know what second messengers do. And you also need to understand what overexpression is, and also how antibiotics work. So, lots of external information was needed to answer this question, but critically, still the most important thing to understand was the passage. You need to know what the GPCR 43 does. If you didn't know what it does, you can't answer this question. Alright, next question. Which conclusion about glucose uptake is best supported by data in figure 2? Alright, so they're straight up asking us to interpret figure 2. And we already made an interpretation at the very beginning about what's probably important. We noticed that the acetate seems to be important for a reduction in insulin sensitivity. And this is only present when the wild type is here. If the GPCR 43 is not here, having the acetate doesn't do anything. So, let's go for the answers. Acetate suppression, insulin-mediated glucose uptake, and wild type, but not GPCR 43 adipocytes. Let's see. Yeah, that's exactly what's happening, right? Acetate suppresses insulin-mediated glucose uptake. So that was the third figure, right? Or the third part of this graph. You saw that the acetate, when it's present, suppresses the glucose uptake. And it's significant with that nice asterisk. But not in the GPCR 43 negative, or knockout adipocytes, which is also what we saw. When it's knocked out, acetate doesn't do anything. If you have it or you don't have it, no change. So this is probably the answer. What about the other ones? Acetate stimulates insulin-mediated glucose uptake independently of the GPCR 43 expressed in adipocytes. So, what this is saying is the GPCR is not important for acetate, and the acetate causes an increase in insulin-mediated glucose uptake. Is that true? No. You can tell from all of these that the presence of acetate doesn't really do anything except in this specific case where it caused a reduction of glucose uptake, not an increase in glucose uptake. So, rule that out. Acetate causes an increase, or causes a reduction of glucose uptake, not an increase. So you can rule this one out. Insulin suppressed glucose uptake in the presence of acetate in wild type adipocytes, but not GPCR 43. So insulin suppresses glucose uptake. Well, this already doesn't really make sense. You should remember that insulin causes an enhancement of glucose uptake. But they are talking about in the acetates. So maybe the acetate causes a reversal of what insulin normally does and causes it to suppress glucose uptake? I don't really think that's likely, but I don't know. Does the graph support that? No. Once again, we don't see any situation where the insulin is getting a suppression of glucose uptake. I mean, you see that the acetate is causing the reduction again in insulin, but it's probably not the insulin that is getting reduced in the glucose uptake. It's probably this acetate molecule, right? Like, the insulin still wants to enhance glucose uptake. You can see. The insulin normally does increase it. It's the fact that the acetate is preventing it. So, not correct logic. And then finally, D. Insulin stimulates glucose uptake in wild-type adipocytes, but not GPCR40 free adipocytes. No. If that was the case, we would only have seen these ones should have been elevated because they have insulin and they're wild-type, and these ones should not have been elevated because these lack the GPCR40 free. That's not what we're seeing. Throw it out. A should be the answer. And it is the answer. And so let's see if the logical reasoning is correct. This is a biochemistry question that falls under the content of principles of bioenergetics and fuel molecule metabolism, blah, blah. The answer to the question is A, because figure 2 shows insulin stimulates glucose. State suppresses glucose in wild type. And, yeah. Okay, so they're basically explaining it the same way I explained it. When acetate is not present, you see that insulin does its normal thing. It causes an increase of glucose. But when the acetate is present, it causes suppression of the glucose uptake. That's significance. Alright, and so here's the next question. Insulin signaling results in phosphorylation of the downstream target AKT, which can be quantified by Western blot analysis. Which graphic shows the effect of acetate administered will likely have on AKT phosphorylations in WAT? What's WAT again? Remember, it's about white adipose tissue. And the muscles of WT mice. Okay. So this is important. We can see that this question is talking about what's the difference between white adipose tissue and muscle. So we need to understand what is the difference between the white adipose tissue and muscle. Well, if you remember, in the passage, they specifically stated the GPCR40 free is expressed in white adipose tissue and is not expressed in muscle or liver. Important, GPCR free in white adipose tissue, not in muscle. So that's a critical difference. The GPCR40 free is in the white adipose tissue, but not the muscle of the wild type. So, what does that mean? Well, we should remember that the acetate is the signaling molecule that causes a GPCR40 free to cause fat reduction. And so that fat reduction is being caused by downstream signaling. And in this case, I'm guessing this is probably one of the downstream signals, the AKT. So AKT is probably only going to be elevated. Wait. Insulin signaling results in phosphorylation of downstream target. Okay, so insulin signaling normally causes a downstream target of AKT. Remember that fat suppression probably indicates that the insulin is being canceled out. We saw that from the previous problem if we go back. Sorry, this problem. We saw from this one that the acetate causes a reduction of glucose uptake. So that's probably indicating to us some sort of fat suppression. So if we go back to that specific problem that we're just doing, it's probably telling us that the phosphorylation of the downstream target is being suppressed when we're administering the acetate, because it should be doing the opposite. The insulin normally causes phosphorylation. We saw that the acetate normally causes the opposite to occur, causes suppression. So this probably is going to cause a reduction of phosphorylation of the downstream target, AKT. So let's go look for something like that. Insulin, okay, these are all the same. That doesn't make any sense. They should not be all the same. Throw that out. All right, B. B, we're seeing what we normally expect. The downstream fat, sorry, they're talking about AKT phosphorylation. But in this case, AKT phosphorylation effectively equates to the radioactive glucose uptake, which effectively relates to fat suppression. So in this case, once again, we see the downstream signal, yeah, it's reduced. When acetate is present, we're going to see less AKT phosphorylation. And critically, we see that acetate, when it's present, doesn't do anything to the muscle. Remember, the GPCR43 is not present in muscle. It's not expressed in muscle or liver. So, acetate shouldn't have any effect on it. It should stay the same. This is probably the answer. But let's just check the other ones to make sure. Now we're talking about the white adipose tissue. There's no effect when insulin acetate's present. This is already not correct. We should be seeing a reduction here. But interesting here, we're saying the muscle is reduced. This seems to be reversed. The muscle, in this case, is the one that's expressing the GPCR43, and the fat is the one that's not. But from the passage itself, we know that's not the case. They have it reversed. This is the wrong answer. What about D? Alright, D is indicating that the acetate is causing reduction of AKT phosphorylation for both muscle and fat. That's not what we're expecting. We should expect something different for only the WAAT. The muscle, once again, doesn't have the GPCR protein, so it shouldn't be affected. Throw this one out. The only answer that makes sense is B. Now, this question, even if you barely understand what's going on, you should still be able to get this answer. Why? Because if you go back to the passage, we know that they're talking about the GPCR43 is expressed in white adipose tissue and not expressed in the muscle. So, the answer that we should be seeing, we should see a change in either only the white adipose tissue or only the muscle. We should not see a change for both of them. So we can rule out A because that one is all the same. Doesn't make sense. We should be seeing a change because the GPCR is only found in one. B shows change in only one. So, possibly the answer. C, unfortunate. We see a change in the muscle only, but not the WAAT. So if you didn't really understand, but you did catch the fact that the WAT and muscle are different on the GPCR43, you should be able to narrow down between B and C. And then once again, D, you see the same change for both. You can rule that one out again because one should be different from the other. They should not be identical. So now we're narrowed down to B and C in that case. And then in that case, you'd have to make your best guess if you didn't understand the logic of what this problem was. But you sure narrowed it down more than a 1 and 4. It's now a 1 and 2. Just from understanding that since one is expressed in adipose tissue and one's not expressed in muscle or liver, you should be seeing different changes. But from our earlier logic, we should have narrowed it down to B being the right answer. And it is. So, this is a biochemistry question that falls under the content category, structure and functions of the nervous and endocrine system, and ways in which these systems coordinate the organ system. The answer to this question is B, because it's shown in the passage insulin signal is blah, blah, the spec we're not expecting WTs. Yep, so this is talking about the same stuff. From the passage, we know the GPCR is only present in the white adipose tissue, so we should only be seeing the change in the white adipose tissue. We should not be seeing a change in the muscle. That's exactly what we saw for answer B. And so that is how you go about answering this question. Notice again, this question, no external information really was necessary to understand. Although you did need to understand that the downstream target, AKT, you need to understand that downstream targets generally are what causes the ultimate phenotypic change, which in this case was fat suppression. But aside from that, this question, you really didn't need to have much external information to be able to answer it. All you needed to understand was what was given to you in the passage. So yeah, this is how you go about answering these kinds of questions. I think that's it for this passage. So let me just check. Oh, no, there's still one more question. My bad. It's 12. 7 to 12. My bad. So, one more question. Compared to untreated wild-type mice, antibiotic treatment of wild-type mice is most likely to result in... Alright, so this is related to one of the answers that was given to us in the previous question, which was question 9. Mice treated with antibiotics, what does it do? And what did I say? The antibiotics, it kills bacteria. What's the microbiota made of? Bacteria. Treating with antibiotics probably is killing microbiota, which is then preventing the short-chain fatty acids from being made, which means that the GPCR43 can't be activated. So, what do we expect to see? Increased plasma butyrate levels? Do we expect that? No, probably not. Why? Because the antibiotic is killing the gut microbiota, and the gut microbiota is what makes the butyrate and acetate. So, if anything, the butyrate should be going down, right? Not up. Rule that one out. A decrease in body mass. Is that what we expect? No. Not once again, no. Remember that, like we said, the butyrate is reduced. What is the butyrate used for? It's used for activating the GPCR43. What is the GPCR43? From the data we saw, the GPCR43 causes a reduction in fat accumulation. So, if anything, a reduction in this should cause an increase in this. So B is ruled out. Increase in volume of adipocytes. So that's basically telling us that we're getting fat accumulation, right? The adipocytes are getting bigger. What are they getting filled with? Fat, most likely. So, yeah, this seems to correlate. It might not necessarily be the volume. Maybe we have an increased number of adipocytes, but this is probably the answer. It correlates with what we expect, but maybe D has a better answer. Let's see. Decrease insulin sensitivity in adipocytes. Maybe? Let's think about this. So, decrease insulin sensitivity. Is that what we expect? No. Why? Let's go back to that nice figure 2. When we have acetate present, it causes a reduction of insulin sensitivity. But what is the acetate made from? It's made from the gut microbiota. And what happened to the gut microbiota? It was killed by the antibiotics. So this is not what we expect. We should see the same insulin sensitivity. So the answer should be C. And it is. So let's look at the answer. This is a biology question that falls under the content category of the structure, growth, physiology, and genetics of public advice. Well. The answer to the question is C is because of C. Producing removed by antibiotic, then GPCR not be activated. Yep. So they're going for the same logic. So we have antibiotic. What does antibiotic do? Kills the gut microbiota. What does killing the gut microbiota mean? The butyrate and acetate can't be produced. What does that mean? The GPCR43 can't be activated. What does that mean? The fact that they can't be activated means that there cannot be the fat reduction that we saw in figure 1. No fat reduction. So, going through this chain of logic, we can deduce which answer is correct, which is there should be an increase in fat, not a reduction. So C. All right. So that is the questions of passage 2 of the section bank. Hopefully this helped you understand the kind of reasoning that's involved with the biology section of the MCAT. You can see that lots of this question is about logical understanding of how to approach these problems. I know, it's a big pain, and if you haven't been doing this a lot, it can seem daunting. But understand that the more practice problems you do for these, the better you'll get at this. The better you'll be able to understand the logical reasoning that's needed for these kinds of problems. And if you ever need help on specific problems, feel free to comment on this post about your specific problem or this specific YouTube video. Feel free to comment on it, and I'll try to get back to you on how to approach that specific problem that you have. I'm also on the Reddit MCAT thread, so you can post there, and there's a good chance I'll see the question, and I'll try to answer it if I understand the question. There's a good chance I will, but remember that I haven't taken the MCAT for I took the MCAT a couple months ago, so since I post this video, so my skills might be a little bit rusty, but hopefully I can help you where I can. Alright, hopefully this is helpful, and if you want more videos like this, comment in the message. It's the first time I made a video in a long time, and I hope it's helpful to all of you. Alright, have a good one.